Okay, can you all hear me? All right, thank you all for being here today. My name's Tracy Lyons. I come from the University of Colorado, where Pearl Scientific is our spinoff. And I'm really excited to talk to you today about our work where we're working toward transforming, is that okay? Okay. Treatments for breast cancers through SEMA-targeted antibody therapies. So despite all of the new therapies for breast cancers out there that you see on television commercials, you hear about, there are very few therapies that are actually curative, and many are just life extending. And so we are really out to find the next target for breast cancer that is durable, targetable, and curative. You may remember over 30 years ago when we found a molecule that nobody had ever heard of called HER2, which now is the result of the best targeted therapies for breast cancer. We are particularly interested in breast cancer recurrence, and we have identified a population of breast cancer patients that have a high rate of recurrence. These are postpartum breast cancer patients. A postpartum breast cancer is defined as a breast cancer diagnosed within 10 years of childbirth. And you may be sitting here thinking, why have I never heard of a postpartum breast cancer? Well, about 15 years ago, we coined the term postpartum breast cancer because we were noticing disproportionately bad outcomes in these patients. And on a side note, postpartum status, or time since last childbirth, is frequently not a part of an oncology patient's chart. So these are not being tracked in the clinic, and we know very little about their biology. What we do know is that they're 3 times more likely to become metastatic, and 25% of our patients will die within 10 years of their diagnosis due to lethal metastases. Postpartum breast cancers are about 150 to 350,000 newly diagnosed patients globally. The reason that is such a large range is because, again, they're hard to track based on the lack of postpartum status in the patient's medical chart. If you could see at the bottom of this page, you would see that 40% of postpartum breast cancers in our current studies are positive for a molecule called semaphorin 7A. But our studies to date of semaphorin 7A showed us that it was more than just a part of postpartum breast cancer. Biologically, semaphorin 7A increases the tracks or the highways that cancer cells use to metastasize. It makes tumor cells resistant to all current therapies, and it allows them to colonize metastatic sites readily while also evading attack from the immune system. So we started to look beyond postpartum breast cancers. And if you separate patients based on estrogen receptor status, or ER here, you can see that whether a patient is ER positive or ER negative, their chances of relapse go up significantly if they are SEMA7A positive. I'm not talking to you today about HER2 positive breast cancers because we already have a very good targeted therapy for HER2 positive breast cancers. So to combat these cancers, we have developed SMAB-H1, which is a monoclonal antibody that targets semaphorin 7A. It not only binds to SEMA on the cell surface, but it also reactivates the immune system to attack the tumor. In numerous animal studies, we have shown that treatment with our antibody results in full regression of 60% of all the tumors. We also observe 0 weight loss and no organ toxicity in response to this antibody. Furthermore, when the treatment was stopped or when the mice were re-challenged with another tumor inoculation, we did not see recurrence of disease, suggesting that we may have a durable and potentially curative therapy. Our mission is to keep families together through all of life's important events. as we heard earlier, being a mom is hard enough. Being a mom diagnosed with breast cancer is just another layer. But it's not just our postpartum patients, it's all women. So to this end, we have developed a first-in-class targeted therapy. We are seeking 10 million in seed, which will get us, hopefully, to first-in-human trials. We have humanized antibodies in hand that we are testing to identify the lead candidate so that we are ready when we are ready for clinical trials. In addition, what I haven't told you today is this is not unique to breast cancer. So SEMA is in about 40% of ovarian cancers, pancreatic cancers, colon cancers, and many brain cancers. So while our lead indication intends to be in breast, we intend to expand as our drug reaches first-in-human trials. We are 100% owned and operated by women, shown here. We also have a unique team of patient advocates who help us to guide our research with the patient in mind. And just as one final note, I will tell you, all three of these women were postpartum breast cancer patients and did not know it until they met us. So another big mission of ours is to raise awareness. They all said, my doctor said my cancer was weird. So with that, I'd be happy to take questions.
Questions?
I have a question. Thank you for the presentation. What evidence do you have to believe this marker is a driver rather than prognostic markers?
We have multiple published studies showing that our antibody will block metastases. We have multiple studies on the biology of the molecule showing it promotes tumor cell invasion, survival in the face of chemotherapy, endocrine therapy, radiation therapy. So we think that it is actually a biological driver of all steps of metastatic and recurrent spread.
Yeah. Do you have patents already in place, or have you licensed this from University of Colorado?
Yes, so we do have 2 patents pending, and Pearl Scientific has the exclusive option to license them at the moment. I am the sole inventor on the patents.
I mean, assuming this works, help me understand who's actually paying for this treatment, because it seems like less of a mandatory thing and a little bit more optional. Is it the hospital? Is it an insurance company? Is it a self-pay? I mean, how are you going to facilitate that kind of adoption?
Well, this would be a novel targeted therapy for breast cancer, so it would not be an optional therapy like HER2. It would be paid for. Does that answer your question?
Kind of. So are you going to try to facilitate adoption? My question is, how are you facilitating adoption?
So our clinical trials will obviously have to start in the metastatic setting in patients whose other treatments have failed them. We eventually believe that this will go to frontline therapy for patients, but first we have to test it in the metastatic setting in clinical trials. And then we hope that, like Herceptin for HER2, this will be a frontline therapy for patients who are positive for the marker. And we are working on a companion diagnostic.
Any questions online?
Can I
Right.
go for my
Go
question?
ahead.
Thank you. So, do you already have an exit strategy or not yet? This is my first question. My second question, can you give us a quick breakdown of the 10 million? How are you going to use them?
Yeah, so took that slide out in the interest of time. But this is kind of how it breaks down with the funding that we have received to date, what we need to get to first in human trials, as well as beyond that, and how we plan to use the funds. We do not currently have a planned exit strategy. No.
What's your frequency of dosing look like?
So in the mice, we have modeled our dosing to be exactly how immunotherapy is administered. So in patients that would be, well, in mice, it's every third day. So I think immunotherapy is something similar to that in humans, but we think we could back off a little bit.
Okay, thank you to Tracy and Pearl Scientific.